Hello. Oops. Anyways, so hi there. My name is Steve Yang, and I'm here to talk to you about what I'm doing this summer in terms of the leadership program lab experience. I'm working at the Baker Institute up on Hungerford Hill Road, which is a really steep hill, and it's really hot sometimes during the summer walking up that hill. But I enjoy it because sometimes I get to pet the horses and like feed them grass. There's like 2 horses there right now on the hill. It's pretty cool. Sometimes people pick me up, which is really nice. Thank you. Okay, so, well, sometimes I also ride my scooter up there. But recently, when I was going down the hill, my scooter, some of the plastic burned off and the brakes kind of just flew off and I burned my hand. Not pleasant, not good. But overall, good day, good time to go up the hill whenever you want, as long as it's not raining. Anyways, so what's my lab? Who's my lab? I'm working in the Van der Waal lab. Most of you don't really know where that is. It's at the Baker Institute, obviously, but it's on the 2nd floor in the new wing, or who she is. So I have a picture of her for you. This is Dr. Van der Waal. She's a leading expert on EHV1, and her research mostly involves the use of stem cells and their various properties to see if they can be used therapeutically outside of just regeneration or wound healing. Currently, she has projects on EHV1, obviously, and mammary breast cancer and hemangiosarcomas. I'm not working on the hemangiosarcoma or the mammary breast cancer projects, which are looking at why horses don't get breast cancer as often as dogs or cats, let's say. Anyways, so what is EHV1? Well, first of all, what can we compare EHV1 to? It's a herpes virus, equine herpes virus 1. And we could maybe compare it to the mandible from Iron Man 3. Wait, sorry, not the mandible, the Mandarin, who is a supervillain. But he didn't always start off as a supervillain. He only became a supervillain because of negligence by Iron Man. He was a brilliant scientist who had this idea for energy conservation or some sort of like crazy energy production idea that would like help humankind. But Tony Stark, obviously being arrogant, disregarded the guy, and he became a supervillain. Tried to blow up the world, take over the world with lava soldiers. The same thing can be said with EHV1. With proper biosecurity protocols, we can prevent the spread of EHV1, which is spread through close contact, respiratory secretions, etc. between horses. It's a virus that can cause mostly respiratory disease, so like tracheobronchitis, rhinopneumonitis, those sorts of things in horses. The same for its, like, I guess, sister virus, EHV4. But there are certain strains of EHV1 that are very bad, and they cause neurologic disease. And the way that EHV1 really causes neurologic disease, and they harp on this all the time, is not by infecting the CNS. Instead, it's through mostly vasculitis. It's a vascular problem that causes both the neurologic signs and abortions. So abortions is the second major problem that a lot of farmers are interested in their mares. And I was going to compare EHV1 to unplanned pregnancies, but I thought that was bad taste because of the abortions. Because it sticks with you. EHV1 sticks with you, or sticks with the horse for life, most of the time, due to its ability to become latent within epithelial cells, primarily expressing only a few genes to keep itself alive and then reactivating during times of stress, such as shipping or whatever. Stressful situations, right? We've all been through those, such as right now, I guess, or 2 days ago. Anyways, so my project is to look at what we call mesenchymal stem cells and their ability to inhibit viral growth in horses of EHV1. Our lab has already shown that mesenchymal stem cells are able to inhibit the growth of viruses within within epithelial cells compared to controls which don't have the treatment. But right now, my goal is to figure out the mechanism by which that occurs. How does it occur? But first, what are mesenchymal stem cells? A lot of you guys are probably asking, what? Aren't they just stem cells? Yes, they are just stem cells, but they are adult progenitor cells that are present in adipose tissue, in cartilage, in umbilicus, in placenta, and in my case, isolated from peripheral blood, separated from white blood cells, red blood cells, platelets, and then you have your mesenchymal stem cells. Ooh, and so far, our lab has already shown that they have also antibacterial properties, which is what I worked on last year, and now I'm on paper on that project. And they also have, obviously, regenerative properties and can be used in wound healing. And our goal is to promote the use of mesenchymal stem cells as an additive effect, besides being able to help wounds regenerate, especially distal limb wounds. They also have other properties as well that they could be used as well for commercial purposes. So what type of samples am I working with, really? So yes, I grow the mesenchymal stem cells in plastic flasks, and after growing them up, we collect what we call the conditioned medium, so the medium in which they're grown in. Supposedly they have secreted factors that then we can use to inhibit the growth of viruses in, right now, corneal epithelial cells or fibroblastic cells. So the second part of my goal is to see if equine corneal epithelial cells are a good model for infection. So far it looks like they're not as good in terms of as an infection model compared to fibroblast cells. And so far for my project, I used these conditioned medium from the mesenchymal stem cells in lyophilized form. So what that really means is that we're cryodesiccating them, we're freeze drying them so that they are in a powder form, and then we reconstitute them. And the purpose of this is mostly commercial, because we are actually thinking that, hey, if this works, this lyophilized version could be used to stall or at least inhibit reactivation from latency or diminish the signs during latency by squirting up the lyophilized version into the nostrils of the horse. And so far, the experiments I've done, some of them, include doing PCR, qPCR, on various genes that I found to be perhaps upregulated during a literature review, so upregulated during viral infection, and to see if the medium might cause the cells to either increase that gene expression or decrease it. And yes, most of these genes are interferon-stimulated genes, obviously. And I'm looking at IL-1, IL-6, GGF-beta, etc. And interestingly, I'm also looking at experiments involving the changes in actin cytoskeleton, because a paper I read showed that EHV1 during infection actually changes the actin cytoskeleton and disrupts it, making the cell less likely to expand and explode, to increase the amount of viral replication within the cell before it explodes, which is very interesting. So I'm staining actin, and looking at cells under the confocal microscope, and hopefully going on to quantification of the cells, which I heard takes a long time. So lots of interesting projects going on now in the van der Waal lab. What did I miss? I don't think I missed anything. If you have any questions, feel free to pose them now. I will be answering all questions from the audience. Someone from the audience can probably answer the questions. No, that doesn't make sense. I forget what I said. That doesn't make sense. I'm not there. Why would they answer the question? Never mind. Okay. Thank you for listening. Thank you for your time. Enjoy your evening. Man, the food looks great there. Bye. Oh, did I show Becky? Becky. So this is Becky Harmon. She's the lab specialist I'm working with. She's smiling here. Pretty cool. I'm not sure what she's working on. The picture's really blurry. I spent like 3 hours yesterday trying to, find a good picture of her. Like I wanted it to be perfect. But this is what I came up with, and I mean it's like all pixelated when I printed it out, and somehow it didn't print from the colored printer. Anyways, so I'm working with Becky in the van der Waal lab. Cool. All right. See you later, guys.